So folks, yesterday I have shared, but somehow the figures, they did not show up. So every time I should click the share. So let us see if today it works. So go to file and then load sequences, your gene pop input data file. And then the gene pop input data file. Here we have the example. Is the example file. So it opens, then click here and save it for the Arlequin. And then standard is good enough. And then go for, then save it. And then continue doing the same for the pop gene. That will have dat extension. And then for pop gene also data set is saved. And then for structure, is saved. So this is how you will convert files for various formats. So let me see if this presentation is showing up really nice. So this time, folks, this is pop gene. So in the pop gene, this is the window once you click the pop gene. As I said, whether it is Arlequin or pop gene or structure, you can download them also, including the convert. So here, go to file and load data. Since microsatellite data is a codominant data that will have both alleles, click that. And then we need to identify where is this data. So the data is in C drive. What is that data called? Example data. So you go to the C drive here, and program files. And in the program files, you have convert files, convert folder. So you need to locate the data. So for that, you need to keep it somewhere, the known place. Okay, where is that? So it is in C drive. So I had two program files. So in one of it, it is there, I think. Yeah, here it is, sorry. And then the pop gene accepts the data in that format that we have already saved. The example file we had converted in the convert presentation that has extension DAT, DAT. So this one you can click, that will show up here. So what is the difference? Any this data formats, whether it is the example file in the GenePop format, or the example file in the Arlequin format, or the PopGene format, or structure format. You can open them in the text format and see the differences, how each program needs a specific data set. So here is POP001. So all the alleles are converted into letters. So the first microsatellites, for first person, the sample is GJ, AG, like that, It's all converted into letters. So only PopGene can understand this conversion. So now we will perform the analysis, go to codominant, deployed data, and here you can say groups, because these are not populations, pretty much groups. And then you can see the allele frequency or Shannon index, especially Shannon index is very important. And then in this PopGene analysis, and the F statistics, okay, and fixation index. So let us select some of these and then say, okay. And then click here, retain all the loci, and then you can just name it also something like that, the number of groups. Whether it did any analysis? No, it did not do it. Let us do it. With the groups, number of groups, probably we should have given the number. So I'm repeating this analysis, deployed data. Okay, let us consider it as a populations because we named it as a population. And allele frequency, Hardy-Weinberg equilibrium we already tested, fixation index, F statistics. And click here, okay. Do you want to retain all the loci further analysis? Okay, keep it. Do you want to retain all the populations further analysis? So it did the analysis. So what we are seeing is for different alleles that are A allele, B allele, we converted them into letters from the sizes, amplicon sizes. Now they are the letters. So A allele, B allele to X allele. And then for each microsatellite, this is the frequency. A allele frequency is this 0.15. In the sense, 10% of the people out of 100 will have this A allele for this microsatellite. For this microsatellite, say 20% of the people out of 100, 20% of the people will have this allele. This is how the allele frequencies can be noted. Like this allele I, for the microsatellite 1, has 70%. Okay, sorry, 7 out of 100. 0.07, it is 7 out of 7 people out of 100. This is 1 out of 100. This is 3 out of 100. So allele frequencies are noted like this. So let us see which is the one that has the highest allele frequency is this one, right? So it is having almost 11 people. 0.11 is, this is 0.23. So these are some very high alleles, 0.25. So 25 people out of 100 will have this allele. So that's how the allele frequencies are important to understand the frequency of that allele in the population. Okay. So the Shannon index is very important. And yesterday also I discussed what is Leventon 1972. Leventon always assumed that eukaryotic populations, especially the species, will have more within species differences than between species differences. This is the Leventon's paradigm. If you want to see the difference between the two different species, that will be lesser comparatively to the individuals within the population. So that's how the eukaryotes are structured. Okay, so this can be estimated using the Shannon information index. So this gives the population how much SSR1 is resolving what Leventon said, what is between I mean within population. So you can see here 2.53 is the Shannon index for this microsatellite, or this microsatellite has 2.52. So that kind of information can be understood. And here is the fixation index. Fixation index is because of inbreeding. If the population has more assortative group, I mean similar types, if the mating between them is assortative mating, okay, so that will reduce heterozygosity. If the population has individuals that are diverse, then the heterozygosity can be seen as excess, lot of heterozygosity. So this is extent of inbreeding, that is fixation index within each population. So this is what you are noting. We have seen the heterozygosity is very high in the Arlequin analysis. That's why here the inbreeding is also very less, except for few of them. Okay, especially this is showing little bit high inbreeding for this one. So heterozygote deficiency can be noted for allele F. So that's how this is interpreted. And then here is it calculates the FIS, which is population differentiation. So this microsatellite is contributing how much population differentiation between the two populations, 0.13 or 0.04, like this, 0.09. So 9%, 6%, 9%, 10%. This is the population differentiation. So this is the, 13%, this is 4%, 1%, this is 9%, 6%. So this much population differentiation each microsatellite is contributing. And then FST, I mean, this is inbreeding depression again. The FST is the population differentiation. So SSR1 is contributing 5%, 2%. If SSR3 is contributing 2%, SSR4 is contributing 2%, SSR5 is contributing 4%. So at the mean is this microsatellite markers that we have used. On average, they are contributing, each one can contribute 3% on average, differentiating between the two populations. So FST is the measure of population differentiation, and the FIS is the measure of fixation. That is that happens because of inbreeding, or the assortative mating. That is, if the same kind of individuals are in the population, then you can see the FIS more. Or if the diverse individuals are there, then you can see the FIS less. So that is how F statistics is interpreted. So this Shannon index analysis we have done. I will record this separately and send it.
ఎవరు లేరు అమ్మాయి బాధపడుతుంది అల్లుడు ఏదో ఒకటి తెలుస్తుంది నన్ను బాధపడుతున్నా కదా నువ్వనే ఉన్నారు కదా బాగా లేరు చెప్పండి.
So students, this time we are running structure for population structure. In Convert, we had created a file with structure format that will have str extension. Okay. So for creating structure, I opened here the structure program. As I said, all of this can be downloaded. And also we loaded it onto your computers, so you already have them. So name of the project, for example, say class. Class is the name of the project. And then we are selecting the diverse directory from where we are doing this analysis. We are doing it in the C drive. Here in the C drive convert. So here we are doing the analysis. So for the class is the project. So I'm selecting this. And now I need to identify where is that str that we have saved, str format. So you see here, example data file dot str. Select that one and then say select. So now it shows this one. The project name is class, and then we selected the directory, and then here we chose the file which with the str extension, and then next. Here you probably forgot how many individuals are there, how many loci are there, etc. details. So you can always verify it. It has 72 lines. So each allele has one separate line. So there are 2 alleles for each microsatellite. So there are 36 genotypes, 36 individuals, and then 7 columns. That means 7 loci. Okay, so here 36 individuals and 7 loci. And always for structure, missing data is -9. That means if the data is not amplified, it counts as -9. So click here next. And then we have the row names, marker names we have. So that I identified it. And then individual population ID I have, like the name of the athlete, whatever is their names, Nolan, John, etc. And then we have their population as in information. And then you can just finish it off. And say proceed. So now you are seeing here data. For example, this guy, Limax, his population is 1. Okay, he's player in population 1. And for first allele for various microsatellites is this, second allele is this. And similarly, here is Nolan. Nolan also belongs to population 1. And this is the alleles for the first allele. This is the second allele. Okay. So you are seeing like that. Here is Hatcher. So for Hatcher, this is the first allele. This is the second allele. And he belongs to population 2. So this is the structure format. Now we will do the analysis. We need to define parameters. Here parameters can be defined, how it has to run, how much, these are the Bayesian statistics. That means you will set everything what you require, that how many clusters are needed or how many groups you want to see, how many lineages you want to see, etc. And how length of the burn-in period and Monte Carlo simulations, how many of them should run. So here is a very simple data set. So I will put 500. If it is bigger data, you can put even 1,000 or 10,000. Or if you want to do some decent analysis, publishable analysis, in the laboratory, we do like 50,000 burn-in period also. It takes a lot of time. So here is the ancestry model. We are using just admixture model. We want to see how each player is admixed genetically. Or you can choose also allele frequency are calculated. That is what we are thinking, because all of them are humans. Some of them are not monkeys, right? So allele frequency model you choose is correlated. And then is there any advanced things you want to do, you can do it. So that is all fine. For this data set, you can just say, okay. And then it asks what is the parameter name. Say we give Vivian. This is the parameter set for all this. So now you have the parameter set, and then you will say run the program. So it is asking how many populations, within population, how many groups or lineages you are assuming. Say I am assuming, say 4 lineages, okay? And then, okay. So the analysis is run. So what is this analysis? Now you can see it. You can click here, and here is the bar plot. You can see it. So now various lineages, 4 lineages we created for each player. What is the extent of that genetic lineage? Say there are 4 lineages in these 2 populations. And this green lineage for this player is highest. He has lesser blue lineage comparatively to the third player, where he has more blue lineage and comparatively lesser green lineage comparatively to him. Okay, so you can see the differences between the players and also the populations. So this is what we wanted to do this analysis. So it analyzes admixture, like you are analyzing data, say Chinese people or African Americans or Africans or Indians or Nepalese. And then you can assume that this many lineages are there, are distributed in this population, and how those kind of lineages are shared. That is what this analysis shows. So some of them will have a particular lineage more comparatively to the others. That's why we want to do this analysis. Okay, so you can save the data, this figure. Also, you can do histogram of say alpha. Alpha is the diversity for various alleles. You can see how the alpha diversity is distributed across the populations, or you can see FST, how the FST is distributed between the populations among the various players. So this is how it is distributed. So various lineages, say the lineage 1, how FST is distributed, for lineage 2 FST, lineage 3 FST3. So this is how you can see lineage-wise population differentiation. Okay, and then you can also see it here. These 2 are the genetically more related populations. Population 2 individuals are green, population red individuals, population 1 individuals are red. You can see how they are clustered in this. Okay, if you want, you can make a tree plot also. So you can say this lineages 1, 4, 2 are grouped together, whereas the third lineage is showing more diverse. So this kind of analysis can be done using the population structure. The plant breeders will do it. This is very important for plant breeders because we have structured populations. The breeding populations are structured, and we would like to know how they are admixed. Like breeding lines from, say, tomato lines from Mexico, tomato lines from South America or US, India, China. You want to see how different tomato populations are admixed. You can understand doing this analysis. And this is very basic analysis for doing association mapping also. So you will use these parameters to control population structure, to control the spurious associations in the association mapping that you have done already. Instead of using PCA coordinates as to control population stratification, if you want, you can use structure. In fact, structure is more preferred. So that's how the structure can be understood. So please listen to these 4 videos separately. First one is convert, second one is Arlequin, third one is pop gene and structure. I have sent you the data sets, analyze them and submit me the lab report. Thank you. So for Arlequin analysis, this is working. So I just recorded convert. So for Arlequin analysis, let us do the analysis. So the file that we created using the convert, we are going to use here for population genetic analysis. So every time I will share it if it is required. And then here, the Arlequin data is here in the convert. So you upload that Arlequin data. And then here you click load into the Arlequin after translation and click translate. Okay. And then go to settings here. And then for AMOVA, for analysis of molecular variants, you need to click here. So this got highlighted. And then you want to do population comparisons like compute pairwise FSTs, you can do that. Or population differentiation, that exact test that tests significance for this FST. And then the Hardy-Weinberg equilibrium, if you want to calculate it, you can do that. And then molecular indices, diversity indices, you can click here and that will be performed. And here, in fact, you can do much more details, a number of different alleles and all. Since the data is not complicated, let us not do it, all that stuff. So this is the one. And then you go to the top of the window. Let me drag this fellow. Here, start. So you click start and you see the analysis. You see the analysis here. So this is what we just performed. So let me share it in case it is not showing up. Yeah, it was not showing up. Good thing I'm sharing it every point. Yeah, now probably you can see it. So here we calculated expected heterozygosity and the observed heterozygosity. And then you see for various microsatellite loci, we have calculated expected heterozygosity for the population 1. So what is expected heterozygosity? Amount of heterozygous alleles that are amplified, amount of heterozygous loci that are amplified. And the p-value is not significant, indicating that they are not deviating from the Hardy-Weinberg equilibrium. So they are all neutral alleles. This data set, as I said, these are some sportsmen in 2 teams of Canada, and they amplified and want to teach population genetic analysis, amplified using the microsatellites. So that microsatellite data we are seeing here for analysis. So here you have 7 different loci, and here is the expected heterozygosity observed as well as expected. This is actually observed using the data set, and this is what we expect from this kind of data if it is not in the equilibrium, Hardy-Weinberg equilibrium, or if it is in the Hardy-Weinberg equilibrium. So we can detect that. So here is basic properties of the data for the population 1 and population 2. Usable data were 6, and usable data for population 2 is 002. So this kind of output will come. So here expected heterozygosity, you are comparing between the populations for each microsatellite locus. Okay. So this all the data is very important. And then you have here number of alleles that are amplified for each microsatellite in different populations. This is the way it is. So that's how 2 populations are compared. And then the most important is analysis of molecular variants, where the variance components can be seen here. So among the populations, between the two populations, the variation is very less, 2.5%. Whereas within population, it is 97%. So this is because the athletes are not genetically related within the population. They are probably coming from different families. They are not related. That's why the variation is very high within population rather than the between the population. So that is what analysis of molecular variance is used. So this is the analysis. And then for each population, you have Hardy-Weinberg equilibrium again calculated, focused analysis. Same thing, pretty much what we have seen up there. There is nothing difference. One thing I did not discuss above is FST that we can even, FST is there here in the molecular variants. Yeah, the FST will be there. FST p-values. Here is the FST. That is the population differentiation. Okay, distance. It's not FST. FST, let us see it in the PopGene format. This has not given properly. Okay, so I will stop here, record this, and then open PopGene. Okay.